Okay. So I will tell you about trace metals bioaccumulation in oyster, Crassostrea gasar from Fresco lagoon. So I will follow the following plan: introduction, material, and methods. We do that in discussion, and at least conclusion and a few perspectives. Trace metals are non-biodegradable, persistent, and are known to cause deleterious effects on animal and also human health. Both acute and prolonged exposure to trace metals cause various diseases. Trace metals also enrich aquatic ecosystem naturally through geochemical, behavioral, or through domestic, agricultural, and also industrial activities. Estuaries are even more exposed to the accumulation of these pollutants, originating from various sources such as atmospheric diffusion, deposition of solid and soil leaching, mainly in agricultural areas, industrial effluent emission, sewage collection, garbage collection, and also mining wastes, and enabling the accumulation of various metals in marine organisms have cultivated or extracted in these environments. Recognizing the potential contamination by heavy metals is especially important as they are extremely toxic and may persist in extended periods and are difficult to remove due to their resistance to reduction and also degradation. Oysters are bivalve mollusks, also organisms attached to underwater substances, where they live and grow, obtaining nourishment by filtering plankton from the surrounding waters. This unique feeding mechanism, while essential for the survival, also renders them particularly susceptible to bioaccumulation, especially of trace metals. Consequently, oysters residing in polluted waters often exhibit decreased concentrations of toxic metals, posing health risks to consumers. Oysters are widely consumed in coastal regions and are integrated to local diet. Food safety is an issue that is very important for public health, as specified by the potent regulation with WHO suggestions and many research studies in the literature. Unfortunately, products from artisanal fishing escape any health control, thus exposing consumers to health risk, and no study has yet focused on the quality of oysters from Fresco lagoon. However, oysters collected in Milliardaire Bay, located in Brun lagoon, so in Cote d'Ivoire, is exhibited high concentration in toxic metals such as lead. A comprehensive survey of trace metals in oysters from Fresco lagoon and their risk to the consumers is lacking. So, in the present study, the concentrations of lead, cadmium, chromium, cobalt, iron, and zinc in oysters were determined, and the potential risk, non-carcinogenic and carcinogenic linked to these metals by consuming oysters collected in Fresco lagoon for children and adults were estimated. The sampling area is shown here in red color. So here is the coordinate. And the study area is located in tropical climate with 4 distinguished seasons: a long dry season from December to April, a long rainy season from May to July, a short dry season from August to September, and finally a short rainy one from October to November. And these seasons are characterized by significant variations in rainfall, continental inflow from rivers, mainly Niñuru and Bolo, and oceanic inflow via the channel, which impact the lagoon's water levels, salinity, and also water quality. During the sampling of oysters, some physical-chemical parameters, such as temperature, pH, dissolved oxygen and salinity were measured in situ using the presented multi-parameter. Regarding oyster sample collection, about 154 oysters were randomly collected at the sediment surface, stored in the original water, and transported to the chemical lab of the Oceanological Research Center of Abidjan. At the lab, oysters were washed with tap water to remove any remaining debris, and the shell length of each individual oyster was measured from the ambo to the farthest point. After shell length measurement, the oysters was spent with a spatula, drained and weighted for weight determination. Then the wet sample was oven dried at 150 degrees Celsius until a constant weight was obtained approximately a few hours, and weighted again for dry weight determination. And finally, each jar sample was crushed in Dagat mortar to obtain a powder, and approximately 1.5 grams of each sample was mineralized in the micro oven in the presence of nitric acid for the determination of trace metal concentrations using a film atomic absorption spectrometer according to standard methods. So here is the spectrometer used for trace metals concentration determination. For health assessment, several indices were used, such as the estimated daily intake, EDI, the total hazard quotient, TQH, and hazard index separately for both children and adults. The estimated daily intake we estimated using the following formula, where MC is the trace metal concentration in oyster shell, IR is the injection rate. So here estimated by in consideration with 30 and 50 oysters consumption for an adult and a child respectively. It also assumes that the body weight values are 15 and 70 kilograms respectively for a child and an adult, according to the literature. Also, the target hazard quotients were estimated using the following formula. In this formula, THQ is the non-carcinogenic risk and is dimensionless. EF is the spatial frequency. AD is the spatial duration, estimated to be 70 years. And RFD is the reference dose of individual metals. And finally, ATN is the averaging time for non-carcinogens. So after the determination of target hazard quotients for individual metals, the sum was used and expressed here by the hazard index, which is the sum of individual THQ values. We are in table 1, we show here the results regarding the physicochemical parameters. The lowest values of dissolved oxygen was observed during the short rainy season, also here called sometimes flu season. The lowest dissolved oxygen values can affect the oysters feeding. pH values were found to be safe for aquatic life, and highest temperature values occurred during the flood and dry seasons in relations with the increase of sunshine. And finally, salinity values were in the following descending range: short dry season, the short rainy one, and the long dry season. In relation with water duration with fresh water that occurred during the flood season. Regarding biotic parameters, the results are shown in table 2. And also, Crassostrea gasar exhibits significant variation in biotic parameters, such as growth rates, mortality, and biochemical responses, often influenced by environmental factors like temperature, salinity, pollution, and food availability, and physical stress responses, combined with temperature and pollutant exposure. Figure 4 presents trace metal concentration in oysters. For lead, chromium, cobalt, and zinc, the following ascending rank was observed for the 4 ones. The lowest values were recorded during the short dry season, followed by the short rainy one, and the highest values were observed during the long dry season. For cadmium, iron, and nickel, the following ascending rank was observed. For cadmium, from short dry season to the short rainy season, and for iron and nickel, from short rainy season to the long dry season. Trace metal concentration values in the oysters depend on several parameters, such as the ability, the nature, the quality of food, metal speciation and concentration, physical and chemical condition of local environments. Now we talk about the estimated daily intake values, and the values are presented in table 4. So for these values, seasonal variation were observed for both trace metals, and the following ascending rank of EDI values were observed for child, cobalt, followed by cadmium, nickel, lead, zinc, iron, and the higher concentration were observed for chromium. And what about for adult? The results are shown in table 4. As for children, EDI values, seasonal variation occurred also. And the same ascending rank were observed for adult, so for cobalt to chromium. Regarding the carcinogenic risk linked to trace metal assessments, the target hazard percent values are shown in table 5 for children. And in a good year to say that for threshold values, the THP value should be lower than 1. So THP value lower than 1 are known to be non-carcinogenic risk. And here, these values were available for nickel, cobalt, iron, and zinc. THP values located between 1 and 5 are low carcinogenic risk, and that was available for cadmium. And a moderate carcinogenic risk was observed for lead with THP value higher than 5. And for chromium, a high risk was observed with THP value higher than 10. For adult, a non-carcinogenic risk was observed for nickel, cobalt, iron, and zinc. A low carcinogenic risk linked to cadmium and lead was observed, and has for children a higher risk linked to chromium was observed with THP value higher than 60. So let's talk about with ash high values now, which is the sum of the THQ individual values for trace metals. The results are presented in tables 7, and all values here are higher than 1. However, the present study values are higher than 10, considered to present a high rates for consumers. Consequently, there is a public health concern linked to the studied oysters consumption in all seasons. What can we say in conclusion? The risk linked to oyster consumption was assessed in Fresco lagoon by calculating the estimated daily intake, the target hazard quotients, and hazard index. Seasonal variation were observed in trace metal concentration, so ADI, THQ, and ash high values. High THQ and ash high values have highlighted the effectiveness of public health concern linked to the consumption of oysters from Fresco lagoon for both children and adults. So there is a need to reduce metal contamination in Fresco lagoon to improve oysters quality and protect consumers from diseases such as cancers, Alzheimer's and Parkinson's diseases, renal failure, decreased learning capacity. As perspectives, we should identify the main anthropogenic sources of metals, notably lead, cadmium, and chromium, and raise awareness among public authorities and population to take action to reduce the contamination of Fresco lagoon waters. And finally, the assessment of oysters contamination with other pollutants such as pesticides due to their use in agricultural activities around the lagoon. And so, thank you to all of you for your attention.